Hi, in this video, I'll be talking about genome-wide association studies in combating COVID-19. The most recent pandemic of the coronavirus disease 2019, also known as COVID-19, is due to the severe acute respiratory syndrome coronavirus 2, also known as SARS-CoV-2. So, GVAS, also known as Genome-Wide Association Study, is a study to identify genetic variants of a quantitative trait and detect an association between single nucleotide polymorphisms, SNPs, and disease in humans. The transmission of SARS-CoV-2 occurs through droplets that were expelled during coughing, sneezing, and talking. If we look at the structure of SARS-CoV-2, it contains positive sense single-stranded RNA with crown-like spikes on the outer envelope surface. The spike protein is responsible for the binding of the virus to the host cell by binding with the angiotensin-converting enzyme 2, ACE2. The uptake of the virus is facilitated by type 2 transmembrane serine protease TMPRSS2 through cleaving ACE2 and activating viral S protein. Both of ACE2 and TMPRSS2 are particularly expressed in lower respiratory tract such as type 2 alveolar epithelial cells. In this study, we'll be recruiting 2,200 patients with symptomatic COVID-19 infection which were hospitalized and tested positive. For controls, we'll be recruiting 2,100 healthy participants. For samples processing, all the samples were subjected to DNA extraction, and then to conduct genotyping, we'll be using Illumina's bead chip microarray technology to assay all of the SNPs. Before analysis, we'll have a quality control, which is important to screen and exclude samples that have high or low heterozygosity rate. Excess heterozygosity rate may indicate sample contamination. Besides, SNP must also be screened and filtered using a call rate of 95%. This is to remove SNPs with more than 5% of missingness. Minor allele frequency MAF threshold can be calculated according to the size of the sample. The last analysis used will be Hardy-Weinberg equilibrium test, which is conducted to exclude the potential genotyping errors and population stratification. In statistical analysis, first we'll be using genotype tissue expression GTAX to analyze the expression of quantitative trait loci for ACE2. Then a Manhattan plot will be generated to display significant and non-significant data points. Logistic regression analysis will be adjusted for sex and age because it is found that ACE2 receptors is located on the X chromosome gene but has been shown to escape X inactivation. Lastly, the Plink platform will be used to conduct a meta-analysis to determine the inconsistency or heterogeneity of the association across multiple GVAS. As we can see from Table 1, the association of ACE2 EQTLs in COVID-19 patients and healthy individuals. The highest MAF in COVID-19 patients is 0.62 and the lowest MAF in healthy individuals is 0.28. As we can see from here, there are 7 SNPs that were associated with increased tissue expression of ACE2, resulting in an increased need for hospitalization due to COVID-19. In contrast, there are 2 SNPs that were associated with a reduced need for hospitalization due to COVID-19. Now we'll be taking a look at the studies that have been conducted by other researchers. According to Chow, Asian populations have higher allelic frequencies associated with higher expression of ACE2 in tissues. This could indicate that differences in allelic frequencies of ACE2 in different populations account for the diversity of expression pattern of ACE2. Moreover, it was also found that the expression of ACE2 in type 2 alveolar cells was higher in Asian compared to African American ethnicity. Another theory proposed by Pan is that ACE2 receptor expression could be elevated in Asian population due to intake of drugs that are mostly prescribed in Asian population. For example, for the treatment of hypertension disease. On the contrary, there was no significant difference in the level of expression of ACE2 in Asians found in a study conducted by Chen, Shen, and Qian. Moreover, there was also no evidence to support that the expression of ACE2 variants when comparing Italian populations with Asian populations. Several in silico studies were largely conflicting is because there are lack of functional studies. However, there is a lack of a specific analysis on the Malaysian population. In summary, EQTL variants in ACE2 may affect the tissue expression of ACE2 in Malaysian population, which resulted in an increased risk of hospitalization. These results provide evidence of the association between ACE2 gene with the risk of COVID-19. However, further genetic studies are required to evaluate the functional role of genetic variation in TMPRSS2, which were the major entry for SARS-CoV-2 to host. These were the references that were used in this video. Thank you.